My pleasure to introduce our first speaker, chemistry professor Matt Francis. Matt received his BS in chemistry from Miami University in Oxford, Ohio, and his PhD in organic chemistry from Harvard. So you can think of him as an Oxford and Cambridge guy, along with a Harvard grad. He was a postdoctoral fellow in the Miller Institute for Basic Research, right here at Berkeley, working with Professor Jean Fouché. Now, research in the Francis Group is focused on the development of new synthetic methods for the construction of nanoscale materials. Matt has built a thriving research program specializing in the development of new organic reactions for protein modifications with the aim of creating new materials for medical imaging and diagnostics, drug delivery, wastewater treatment, and light harvesting energy systems, among other applications. Matt has taught a broad range of classes, including physical organic chemistry, chemical biology, transition metal chemistry, and freshman-level quantitative analysis, among others. He also brings his love of organic chemistry and his love of cooking, along with his excellence in teaching in his role as faculty advisor for Decal Chemistry 98, the chemistry of cooking. And it's a real treat to sit in on that. And speaking of teaching, Matt received the Campus Distinguished Teaching Award in 2009, the Noyce Prize in Undergraduate Teaching in 2006, and the Departmental Teaching Award in 2005, 2008, and 2014. Oh, and by the way, Matt was the college's Executive Associate Dean, the dean's right arm, if you will, for 4 years, before stepping into his current role as chair of the Department of Chemistry. So please welcome Matt Francis. His ChemX talk today is titled Using Chemical Biology to Develop Clean Water Technologies. Matt.
Okay, thanks, Doug. Thanks for the introduction. Thank you all for coming this afternoon. It's a real thrill to be here and kick off what I think will be a great new event for the college. So as Doug said, my lab works at an intersection of organic chemistry, materials science, and chemical biology. And the primary research activity in my lab is the development of new chemistry that can be used to bring biomolecules together with synthetic components. And we seek to do this for a variety of applications that take the function of proteins, maybe it's catalysis, binding, energy transduction, and put those into a context that we can process, that we can handle, or that we can read out for a specific signal. And so we do this in a variety of areas. Doug mentioned those in the introduction. So they do include a lot of drug delivery materials. They include a lot of materials that are useful for asking basic questions about how photosynthesis works in nature. Today I'm going to talk about some of our most rapidly growing projects in the lab, which are using these protein-based materials for environmental applications. And so just to start out, I want to begin with a reminder of how fragile our water supply is, especially here in California. That what you're looking at here is Lake Oroville. This is the second largest drinking water reservoir in the state of California. You'll see this if you go up toward Mount Shasta. It's up in Northern California. This is that reservoir in 2011, and this is the same picture, that's the same bridge taken in 2014 at the peak of the California drought. And what you can see is a drastic change in the volume of water. Of course, then after this, we've had more rainfall, more snowfall in the Sierras, and this has increased back again, but this is fluctuating back and forth. So while this is happening, and while this is going to continue to fluctuate because we're increasing our water usage, we have more changes due to climate change in the water distribution, we are continuing to increase our activities in agriculture and manufacturing, and also forest fires are becoming more prevalent. And these are introducing more contaminants into our environment. And the point of this is that at times they're being concentrated in much smaller bodies of water. And so the concentration of these pollutants is going up quite drastically, and there are a number of these that are of current concern to the state. So the class I want to talk about today are some of the most difficult pollutants to deal with, and these are the endocrine disrupting compounds. These are compounds all in the family related to the estrogen molecule up here on the upper left. All of these compounds bind to the estrogen receptor and the aryl hydrocarbon receptor. Both men and women have these receptors, albeit in different locations and different expression levels, and this leads to biological effects of these molecules. And so some of these are drug molecules, so this is the birth control molecule here. here. Some of these are drugs or intermediates. Some of these are plastic materials like BPA, BPB, and these are also historical pollutants, including DDT and PCBs. And when they bind to these receptors, it's known that they cause a lot of deleterious effects for fish and wildlife. There's mounting evidence that these effects will also be prominent for humans, but the data are still being analyzed. And so what makes these so difficult to deal with, first of all, is their concentration at which they are active. These are active in nanomolar and sometimes sub-nanomolar concentrations. And to put that in perspective, if you take estradiol, if you take a portion of that the size of a tiny Tylenol caplet that you might take for a headache or something like that, to get the active concentration, you would crush it up and dissolve it in an Olympic swimming pool, such as the one from London here. And so at this vanishing low concentration, it is virtually hopeless for an adsorbate like activated charcoal to absorb those materials and remove it from the water. Secondly, these aren't found alone, that when they're found in nature and groundwater, they naturally occur along with humic acid. Humic acid comes from lignin, naturally from plants. That's this polyaromatic material here. This binds to those absorbates as well, and thus prevents the binding of the much lower concentration molecules of interest. And so really, there is no way to remove these very well yet, and it's even difficult to even know that they're there. And so what we thought is we could use chemical biology to address both of these questions, first as a way to detect these molecules at their active concentrations in water supplies, and then secondly, adapt this technology for removing them once you know that they're there. So we focused on the very proteins that make these molecules have effects on humans. One of them we've worked with is the human estrogen receptor. This is the soluble domain here. It's a beautiful, famous protein in biology. It's got this alpha helical design here. And what you can see is that it has a binding pocket shown here in red for estradiol itself when it binds to the center of this protein. And when it binds there, this green helix comes from the top side and flops into place, just flops right on top of it. And that is the key signal. That change in conformation is the signal that causes the downstream signaling events, gene transcription and the like, that leads to the biological effects of this receptor. And so all of these molecules bind this, and the first thing that we noted is that these have the same, these binding constants are actually at their toxic concentrations or their effective concentrations. And so this would give us the affinity that we needed to actually be able to efficiently bind these. Secondly, in terms of detection, in the literature, the Coida lab, this reference here at the University of Chicago, had developed a really interesting protein called a monobody. That monobody, shown in yellow on the left, binds to this receptor only when this green helix is in place. In other words, this interaction between these proteins only takes place if one of these endocrine disrupting compounds is in the active site binding pocket. And so what we thought is we could turn this, first of all, into a chemical sensor, where we could actually put these on two different components of a sensor, and we would have something that we could take into the field and in real time quantify whether these compounds are here, and we had a hope that it would actually have the sensitivity and selectivity to work in a real environmental sample. So to do that, this is our platform. This was developed by Ariel First and Alex Hufker in the lab. And what they did is that they split this into the components. They first put the monobody here on a gold surface. That's one of the binding pieces. And then, rather interestingly, they genetically fused the estrogen receptor to surface proteins on E. coli. So you can grow E. coli in the lab. You can grow this in any quantity that you want. And each of these bacteria, as we produce them, have about 20,000 copies of the estrogen receptor, shown in blue, on their external surface. And we take that sample, we isolate the bacteria, and we freeze dry them. That renders them then non-viable. It also makes them so you can store them. Okay? So then the way this should work is if we have a sample with an endocrine disrupting compound, this will form a sandwich complex here to bring these together, and therefore the bacteria will engage the surface. And you can actually see this. These are electron micrographs. On the left is a polycrystalline gold electrode surface, and there's E. coli in there, but they're not engaging that surface. But when you add a sample of estradiol to it, you can now see the bacteria clearly binding to the surface here. And so that confirmed to us that this interaction was actually possible for putting the bacteria down on the surface. Now, when the bacteria come into contact with this surface, that's going to cause a lot of changes in the ions that move around across the surface of this. And that allows us to use electrochemistry to detect when this event has occurred. You can think of a lot of ways to do this, but the way that we set this up is, first of all, here, where we make these electrodes. They're about $2 to $3 a piece. I actually brought one. They're about this big in real life, so they're quite a bit smaller than a microscope slide. And the active place where we have our monobody is this circle in the middle, which is about the size of Roosevelt's ear. And so then what you do is you take a water sample that contains maybe endocrine disrupting compounds, you take the bacteria, you mix them together, and you put a 10 microliter drop on that dot, and then in real time you use electrochemistry to see if the engagement occurs. And so, so here's how we do it. On the left, this is called a Nyquist plot. this is plotting the imaginary and the real components of the impedance. As you increase the concentration of estradiol, going down these colors here, you can see this arc increasing in its diameter, showing that these dots are moving farther and farther out. What we can then do is do a circuit fit. We do a circuit modeling fit, which are the solid lines that you see here, and from that we can pull out a resistance, and that is what we're plotting on the y-axis of these plots. And what you can see is if you plot that resistance as a function of estradiol concentration, we get the black dots of data here, showing that we get a linear response from the device. And we're really excited about this for a lot of reasons. First of all, this is, to the best of our knowledge, the first portable device that could reach this level of sensitivity. This not only works at micromolar concentrations, but all the way down to sub-nanomolar concentrations. The actual biological activity level, our detection limit is about 250 picomolar with this system, which means 10 bacteria are actually binding to the surface, and we can still detect that. The second thing about this is that this should actually respond to all of the different endocrine disrupting compounds that are present. Anything on the previous slide can bind this and trigger this system. And so not only one thing could we detect, but we could detect them all, which would be very useful in just knowing if a sample of water is contaminated or not. And so this is estradiol, but you can also see here that this is for nonylphenol. You get a linear response. These are all being scaled versus their IC50 values, which go up and down for the receptor. You can see for genistein, we get a nice linear response. Diethylstilbestrol gives a linear response down to a certain part and then flattens out, but this is down in the low 100 picomolar level. And then the selectivity is maintained for progesterone, a molecule that does not bind to this receptor. We see no response. And so this shows you, again, the power of the selectivity that this has for just the right molecules that engage this biological pathway. So it works for all of these molecules. There's several others that we've surveyed over the years, but perhaps the one of most consumer interest would be BPA. BPA, as I mentioned earlier, is a compound commonly found in plastics used in kitchens and baby products and everything. And so we can see with the cyan dots, we get a linear response for BPA as well. It does bind the receptor, that was already known, and it triggers the system to give a response. What's interesting is that you can do this in a more complex background. We can also do this in infant formula. So if you take infant formula from a commercial supplier, you add BPA to it, you can see these purple dots, seeing that we do have, again, a linear response down to about the KD value. Below that, this levels off, and we actually don't know why this doesn't go down to a lower value. It might be because the proteins interfere with the binding. That's a possibility. It's also possible that there's some of these compounds that are present in the formula to begin with, so we don't know which it actually is in this case. But what this does is it speaks to the ability to use a very complex formula, now something that's opaque and white and everything else, and still get a useful signal. So as a father of a 6-year-old, then that led Ariel to run an experiment that really does keep me up at night. And so what you can do is you can take 2 baby bottles. You can take one made of glass, one made of plastic. You can put formula in those bottles and you'll measure a certain level of background activity here. And then if you microwave those 2 bottles, there it is. Comes right out of the plastic bottle into the formula. And our system is sensitive enough to actually detect that as the small amount is leached into the formula and becomes bioavailable. What's also alarming about this is that this is a BPA-free plastic bottle. It turns out that the BPA isn't in there most likely, but the plasticizers and plastic materials that have replaced it are just about as bad. And so this is probably BPS in this particular case. And so again, shows the sensitivity and the sort of value of having the real-time ability for this device. In terms of then going forward, I think we have a new platform we can use for all kinds of different applications. You could imagine engineering this for many different types of responses. So one of them is to re-engineer this binding interface to go after things that are toxic to the thyroid gland. There's a lot of other compounds we're thinking about building into this system. We're also trying efforts now to re-engineer this to have the surface directly engage the bacterial cell wall, with the idea that you might be able to use this to detect or possibly even speciate different pathogenic bacteria in real time. So we're trying to develop this as something that could be very useful for monitoring the food supply, for example, looking for bacterial contaminants, or perhaps something for use in hospitals. So finally, one of the main areas of interest in my lab is how do we work out the chemistry then to take these materials and put them into large matrices, things that you could filter. And so we've done a little bit of that. This is ongoing work in the lab. This is one example here where this is another protein that binds many of these compounds. This compound is called uteroglobin. It's also known as the PCB binding protein because it has nanomolar affinity for many different PCBs. And we can produce this rather easily. It's a very simple protein to produce. You can get pretty good quantities of it. And we've developed some chemistry in the lab, some very easy low-cost chemistry based on these pyridine carboxaldehydes that can modify just the N-terminal sites, the two orange sites shown at the top. And so you can use this to functionalize this protein, to attach polymers to this protein. One thing that Henrik Munch did is he used this to attach this protein into a sort of gel matrix, something that you could use as a little bit of a filter. And then what you can do is you can pass water through that filter, starting out with water contaminated with PCB70 at about 36 parts per billion. If you pass that water through the filter containing uteroglobin, you can see it's practically all reduced below 5 parts per billion, which is the EPA safe drinking water limit for this particular compound. And so I think the most important result, though, is that you can repeat this experiment with 50 mg per mil of humic acid, and you see no reduction in the binding ability of this, showing again the power of the selectivity that these biomolecules have. So what we're excited for going forward is it's a really good time, I think, for protein materials because we become very good at making large quantities of materials at very, very low cost. This comes from everything from the detergents industry to the biofuels industry. There are ways to make very large quantities of these proteins. Secondly, the polymer materials are becoming more and more available, and what we hope to contribute to this is additional examples of chemistries that can be used to do this at very low cost to make these well-defined materials. Okay, so that's all I have time to talk to you about today, but I want to end by thanking a really terrific group of students and postdocs and undergraduates. This is a nice time to do it on the home turf. They're all over in Latimer 7 over there, so this is the current members of the group on the left side. People who did the work that I specifically talked about today are Ariel First, who's a postdoc who did all of the electrochemistry. Alex Hufker is the one who did the initial fusion of the protein to the E. coli surfaces. And then several people in the lab have worked on uteroglobin, but Henrik Munch was the one who first cloned the protein and made the material that I showed you. Finally, I'd like to thank all these groups for funding. All this work has been funded by this grant from the National Science Foundation, which funds a lot of our work to make new types of water technologies. Finally, thanks again for the invitation to be here this afternoon, and thanks for the introduction.